All right, our first speaker for today is Taylor Abraham. She will be starting her third year of veterinary school at the University of California, Davis, with a focus in aquatic animal medicine. She recently completed her Master's of Preventive Veterinary Medicine at UC Davis under the guidance of Dr. Esteban Soto. Her thesis was completed with Dr. Soto and his team at the Aquatic Animal Health Lab. Outside of studying and work, you can find her surfing or hiking with her dog. And today, Taylor is going to be talking to us about the detection of Lactococcus species in environmental samples and wild fish from 4 lakes in Southern California. So take it away, Taylor.
Thanks for the introduction. So as she said, my name's Taylor, and I've been working with Dr. Soto in his lab for the last couple of years, looking at Lactococcus in environmental samples and wild fish from lakes in Southern California. So to start us off, I'll be talking about the disease we've been looking at, and this is piscine lactocophicosis. And this is a bacterial disease that affects freshwater marine fish and has been found all around the world. Historically, it's devastated trout hatcheries and caused large economic losses when it is found in facilities. Affected fish, most often rainbow trout, will present acutely with a septicemia, as well as also see clinical signs of lethargy, erratic swimming, exophthalmia, anorexia, skin pigment changes, but most often we'll see death. The image on the right is demonstrating some of the pathological signs that we'll see in fish that will present with lactococcosis, especially rainbow trout, such as in the upper left corner, we see exophthalmia, we see internal hemorrhaging in the middle photo. In the bottom left, we see pale gills affecting this fish, and in the bottom right, we see hemorrhaging in the brain. So up until recently, Lactococcus garviae has been the main agent from the Lactococcus genus that causes this disease. L. garviae was originally classified under the Streptococcus genus, but through sensitive diagnostics, it's been reclassified under its own genus, which is Lactococcus. L. garviae has been known to be an emerging zoonotic pathogen. We've found cases isolated in humans, so domestic animals, livestock, as well as been isolated in various environmental sources. But up until recently, we've had 2 new species from the Lactococcus genus that have been isolated, now identified as causative agents of piscine lactococcus. And these are L. petuari and L. formosensis. And both these share a significant portion of their genome with L. garviae, and it's been shown to cause similar clinical signs in fish. So these 3 Lactococcus species are gram-positive, catalase negative cocci, and they have similar biochemical properties, so it can be very difficult to distinguish between these 3 without further molecular diagnosis. So in the past decade, we've found piscine lactocophicosis in 4 states, Canada and Mexico. There are articles on the right just demonstrating its emerging prevalence in trout hatcheries and its huge economic losses that have also resulted from these outbreaks. The origin of these outbreaks is still unknown, but it has warranted concern as future outbreaks and new strains are continuing to be discovered in other regions in North America. Most of the work has been done on L. garviae, as I mentioned, since this is the first one that was identified. And this bacteria has shown to have a high incident rate of disease in trout when they're housed in water temperatures higher than 15 degrees Celsius. So often in the summer months is when we're seeing Lactococcus outbreaks. But as climate change is increasing our global temperatures, we are seeing seasons and water systems with higher temperatures for longer periods of time. So this is leading to a longer transmission period of this disease. So this brings us to California, where we had our first outbreak of Lactocophicosis in 4 trout hatcheries in the spring of 2020, located in Southern California. This outbreak, it did result in the euthanasia of 3.2 million trout, and the isolates are originally identified as L. garviae. We still don't know where the origin of the strain is from, but it has shown to be highly virulent in rainbow trout that we challenged here at UC Davis. It was also shown to be very difficult to treat amidst the outbreaks. So recently in our lab, researchers sequenced and compared the housekeeping gene DRase-B of several L. garviae isolates that were collected from disease hatcheries over the past 5 years. And this revealed that the emergent isolates that affected the trout in California are in fact Lactococcus petuari and not Lactococcus garviae. Historically, the strains that were identified as L. garviae through biochemical and molecular testing are now found to have been re-identified as L. petuari or L. formosensis from L. garviae, as seen through this phylogenetic tree. So this recent reclassification demonstrates the need to further understanding of the Lactococcus genus, as most of the L. garviae strains are now being re-identified as either of these 2 species, and there still does exist a possibility of more species existing. So as you can see here, we have our L. petuari on the upper portion, Lactococcus garviae in the middle, and L. formosensis in the bottom. And the species that these have isolates recovered from range from rainbow trout to catfish, ducks, cheese, and even processed meat. So this outbreak in California in spring 2020 did lead to many questions on people's minds, such as where the strain originated from, what is the distribution of this pathogen in the environment post-outbreak, what species of fish might be infected if we do find it in the surrounding environment, if we find wild fish with the bacteria, can they pose a risk as a carrier, and of course, what is the virulence of the recovered isolates in trout and other species of fish. So all these questions led to our project, which aimed to complete a surveillance of this outbreak in 4 lakes near an affected hatchery in Victorville, California. Doing so, we wanted to better understand the distribution of the bacteria in the surrounding environments and wild fish post-outbreak. Also, we wanted to compare the virulence of these North American isolates of L. garviae, L. formosensis, and L. petuarii in 2 fish species. In doing so, we wanted to gain insights into the host susceptibility and strain pathogenicity. Okay, so to start us off, I'll go over objective 1. So this started in September 2020 when California Department of Fish and Wildlife, they went out and collected wild fish ranged from 8 different species of fish and environmental samples from 4 lakes in Southern California, which was near one of the affected hatcheries. So as you can see here, we have one of our affected hatcheries, which is Mojave River Hatchery, have our 4 lakes. So we have Horseshoe Lake, Spring Valley Lake, Victor Valley College Lake, and Jess Ranch Lake. And our environmental samples were collected from 5 different locations around each lake. So Mojave River Hatchery, it did have water canals connected to Spring Valley Lake and Horseshoe Lake, but no known connections or outlets to Victor Valley College Lake and Jess Ranch Lake. We collected 359 fish, and those were taken at University of California, Davis, where we examined them for any clinical signs of disease and morbidity. We necropsied them and took tissue samples of the spleen, kidney, liver, brain, or the whole body if it was under 1 gram, and we did so for further evaluation and diagnosis. After the necropsies, we extracted total DNA and purified the tissue and environmental samples. And we tested for the presence of L. petuarii, L. formosensis, or L. garviae in the genomic DNA from the tissues and environmental DNA from the soil and water samples. And we did so using a recently developed TACMAN-based qPCR assay that is specific for these 3 species that was here at the lab at UC Davis. Unfortunately, our qPCR test cannot distinguish between the 3 species just due to how highly genetically similar the pathogens are. So we labeled fish who had positive detection as Lactococcus species positive, or L. garviae, L. petuarii, L. formosensis positive. For all of our qPCR tests, we looked at a cycle threshold value of 36 in all of the 3 replicate wells as a positive, and if a CT of 36 to 40 in at least 2 of the 3 wells was considered suspect or weak positive. We also took swabs of the brain and tissues from each fish, and we inoculated this into tryptic soy agar supplemented with 5% sheep blood, and then we incubated for 96 hours at 30 degrees for isolation. And we did further catalase testing and gram staining for further isolation and diagnosis. So for our statistical analysis, we looked at the frequencies of positive samples of the soil, water, and tissue among the 4 lakes using a Fisher's exact test. And then we calculated prevalence values per lake using the following formula at the bottom. So for our results for this portion of the project, we did isolate Lactococcus garviae from the brains of 2 largemouth bass from Victor Valley College Lake. These fish additionally did test positive for detection of Lactococcus species through the qPCR. Through our qPCR test, we did detect Lactococcus species in 14 fish, and these were 8 bluegills and 6 largemouth bass, and they are from 3 of the 4 lakes. So as you can see here, we have most of our bluegills coming from Horseshoe Lake, and then 1 from Spring Valley Lake, and then largemouth bass also from Horseshoe Lake and Victor Valley College Lake. As you can see here, Jess Ranch had no samples test positive. Horseshoe Lake had a prevalence of 6.3%, followed by Victor Valley College Lake with 5.1%, but we did see no statistical difference between the prevalence of positive tissue samples between these 2 lakes. For environmental samples, all 4 lakes did test positive for Lactococcus species in our soil samples, while only 2 of the 4 lakes tested positive in the water samples through qPCR. So as you can see here, Jess Ranch had the highest prevalence rate of Lactococcus species in the water with 100% detection, and then 2 out of the 5 testing positive in the soil samples. Horseshoe Lake followed with 4 out of the 5 for the soil samples testing positive, and then 1 out of the 5 for the water. Spring Lake and Victor Valley College Lake, as you can see, had no samples test positive, and only 1 out of the 5 soil samples test positive. So for our next portion, we want to look at the virulence of these 3 distinct species of Lactococcus. So to do so, we challenged rainbow trout, which are known hosts for the pathogen, and largemouth bass in a laboratory controlled challenge here at UC Davis. We chose to challenge the largemouth bass since they are the only species that had a positive isolation of the bacteria from our wild fish collection. So in our phylogenetic tree, we have our 23 isolates bolded, and these were the ones we used for the challenge. So we had 17 isolates of Lactococcus petuarii, including ones that were recovered from the outbreak in the spring and summer of 2020. And then we also had our L. garviae isolates, and those two were recovered from our largemouth bass. So those were VC11 and VC13. And then we also had 4 of our Lactococcus formosensis isolates. So to start off our challenge, we housed our fish at the Center for Aquatic Biology and Aquaculture at the University of California, Davis. We challenged our fish in groups of 10 fish per tank per isolate to whichever unique isolate of L. garviae, L. formosensis, or L. petuarii they were assigned to. We anesthetized the fish with MS222 and then injected them with 0.1 mils of whichever distinct isolate by intracellular injection of the ventrum. And we also had a control group for each species that was injected in the same manner, but with 0.1 mils of sterile saline. Following the injections, we monitored the fish daily for any clinical signs, mortalities for 14 days. In the first 5 mortalities of each tank, we cultured them to test for the presence of the Lactococcus species, and then the final survivors to 5 were cultured at the end of the challenge period. So again, for our statistical analysis for this portion, we used a Kaplan-Meier survival analysis to look at the cumulative mortalities and survival percentages for each isolate and then for each species. And these images right here, just highlighting some of the clinical signs we saw in our rainbow trout, especially in the first few days of the challenge. So as you can see here, we can see exophthalmia up in the upper 2 photos, as well as skin pigment changes. In the video right here, this was taken in the first couple of days, is just highlighting the acute changes we saw, such as you can see some mortalities already at the bottom. This fish right here, you can see that there's exophthalmia and skin pigment changes as well. So for our results for this section, we did see significantly greater disease severity and mortality of piscine lactococcosis and our rainbow trout groups challenged with distinct isolates of L. petuarii in comparison to our rainbow trout groups exposed to L. garviae or L. formosensis. As you can see here, our L. petuarii groups, they did end up with a cumulative mortality of 62.2%. And as you can see, most of the mortality rates did exceed over 50%. And this did occur in the first days of the challenge. While our L. garviae isolates in the rainbow trout, they had a 0% mortality rate. And then our L. formosensis, only one had any mortalities, and overall it was a 10% mortality rate for the group. We also tested for bacterial resistance at the end of the challenge. We saw no significant difference between the species for bacterial resistance and the surviving fish. And all of our negative controls did remain infected, and no mortalities occurred throughout the study in that group. For our largemouth bass, we saw no clinical signs of disease or mortalities throughout the entire 14 days, and only one from the L. petuarii group did test positive through bacterial isolation and qPCR at the end of the challenge. So in conclusion, we had 2 isolates recovered from largemouth bass, and they were genetically distinct from those recovered during the outbreaks, and this did confirm genetic heterogeneity in the species causing piscine lactocophicosis. This was from Victor Valley College Lake, and it was the only one to have a positive isolation of bacteria. However, it had no water samples to have positive detection of the bacteria and only 1 soil sample. These 2 isolates also had a 100% survival rate when challenged in the largemouth bass and our rainbow trout. But however, the isolates that we recovered from the outbreaks in the summers, they did have a very high mortality rate in our rainbow trout, but no effects on our largemouth bass. We did detect the bacteria in only 2 species of fish out of the 8 species collected from our wild fish collection. And this was the first time that we've confirmed detection of Lactococcus species in these 2 species. Species that are historically known to be affected by Lactocophicosis have ranged from crustaceans to now tilapia, catfish, gray mullet, and it's even been found in some marine mammals. So finding this species, finding Lactococcus species in these 2 species does show some host association in the bacteria, but they did show no clinical signs or morbidity upon our exams on them. Finding positive detection in wild fish can be worrisome since wild fish can pose a carrier host risk, and this can lead to transmission of the pathogen if conditions are conducive, such as during high temperature times. This study was also able to demonstrate the potential use of qPCR to detect eDNA of L. garviae, L. petuarii, and L. formosensis in the environment. It has been shown that L. garviae and L. petuarii are able to form resistant biofilms and persist in the environment unless treated with specific detergents. And it has been proposed that transmission through contaminated water, soil, and fecal contamination by birds can be mechanisms of transmission for these pathogens. Thus, being able to use eDNA as a useful tool for detection of these pathogens in the environment can help further our understanding of the prevalence post-outbreak in surrounding environments, and also hopefully this can help inform management of hatcheries on ways to prevent outbreaks and predictive spread in the environment. Additionally, we're able to confirm that Lactococcus fari is a highly pathogenic emerging pathogen of rainbow trout in the USA. So thank you to Dr. Esteban Soto and Dr. Eden Gardner, the amazing Aquatic Animal Health Lab team at UC Davis, with all their help, especially through the qPCR, CDFW for all their help, and our collaborators across these universities and facilities. Any questions?
Thanks, Taylor. That was a great talk. Yeah, we have time for questions, so go ahead and feel free to raise your hand, unmute yourself, or drop your questions in the chat box.
Hey Taylor, I'll jump in here. Great talk. I really enjoyed that. I apologize. I logged in a little late. Do you have any idea of the validity of the sequences of these Lactococcus garviae in GenBank? It seems like things could be probably a mess based on some of your trees. And what advice would you give to people when they come across how do you get a proper identification? What would you suggest when you encounter these bacteria?
Yeah, currently in our lab, they've been doing MSLA and whole genome sequencing. So I think that is the most specific way now to identify which strain might be infecting certain hatcheries or fish. I don't know if that helps answer your question.
Yeah, I mean, I guess to that end, but as far as what's available in GenBank, how much of a mess are we in here with these isolates? Everything seems to be identified as garviae. How much of that is accurate, would you say? And you may not know the answer, and that's perfectly okay.
I know they are currently still reclassifying a lot of them because, as you can see, like the phylogenetic tree, a lot of them are still identified, yeah, as L. garviae, but I think they're still working on it currently in our lab right now on working on ways to reclassify them as different names, just due to this whole mix up now, and there could be other species that we just don't know about yet. So sorry, to answer that question, I think it's still being worked on right now.
Yeah, I just see in your tree, you've got all these ATCC, those are type strains, but they're obviously incorrectly identified. So
yeah. Go back to it.
Yeah, it's a mess.
It is.
Great talk, Taylor. I really enjoyed it.
Taylor, I have a question for you. If I'm remembering this right, when you were doing the sampling of the lakes, there was quite a big difference, especially with, I think it was Just Lake, the fourth one, maybe, that had no positive fish, but had the highest number of positives for your environmental samples. What do you think factored into that variation across the fish versus environmental in the 4 lakes?
Yeah, and there is actually a private hatchery located near Just Ranch Lake, and it's called Just Ranch, and they did also have an outbreak there. We just didn't sample any of the lakes surrounding that area because we focused on the Mojave River hatchery. So I think that is why we had the water samples test positive, but having no fish, I don't know. It is interesting that we had none, but fully 100% for the water and almost all the soils.
So you have a question in the chat. With your data from different samples and sample types, have you considered using occupancy models to handle some of the false negatives in your prevalence estimates?
I have not. That'd be an interesting thought, an idea, something we can look into.
Any other questions?
I have a short question. So if there is some concern with the broodstock, is there any recommended improvement for their management given potential or some substantial latent infection of the virus?
Sorry, could you repeat? Sorry, could you repeat that?
From your study, because there are some broodstock hatchery that they might have some latent infection. What do you think that the suggestion or any improvement for the broodstock that they can cope up with a latent infection?
Yeah, it's a great question. I know they've made some vaccines, so I think vaccinating is the first way to prevent any outbreaks, and then of course just monitoring your brood stocks and doing testing probably often to see if it is latent, which you can do through biochemical tests.
Okay.
Good.
Thanks. Thank you.
Yeah.
Yeah, just to add a little bit into that one, Taylor's absolutely right. CDFW at least, have been pretty aggressive into testing any facility that has come positive, for this bug. And there has been vaccination, started since last year. Both immersion and intracellular injections have been now established in most of the hatcheries and the private aquaculture facilities that have tested positive. And before they release, the animals, they have to show evidence of negative isolation of the bacterium.
Thank you. Thank you.
Any last questions?
I can ask one, Taylor. Why didn't you sample any salmonids?
Good question, Dr. Soto. We didn't sample any salmonids because we weren't able to collect any during the collection time, which was in the summer. And primarily salmonids, they will go to deeper waters or cooler waters during this time since they prefer the cooler temperatures. So there could have been some in the lakes, but they weren't present during that time and throughout the collection. So we're unable to confirm if any salmonids did have any positive detection of the pathogen throughout this time.
Thank you, Taylor. Yeah, and I just wanted to bring up that point, because it's an important one to consider, as this bug does seem to have a pretty high host association with trout. So the fact that some of these lakes, again, they aren't made, for having trout. They get pretty warm during the summer, so most of the trout, wouldn't even survive in there. But the persistence, risk, comes in because if they're deep enough, some of the salmonids, could actually go there, hide during the summer months, and then establish, persistent infection, as the bacteria can persist when the temperature is lower. And then, there is risk that in the following year, once the temperatures increase again, there will be shedding of the bacteria. So it's one of these bugs that we have to associate really clearly with environmental conditions.
All right, thanks, Taylor. Everyone want to give one final round of applause?